Hello. In this video, we are going to discuss about microsporogenesis, which is also known as microgametogenesis. This topic is part of chapter number 2, sexual reproduction in flowering plants. You're shown here with a part of stamen. Stamen is male reproductive organ of flower. So flower is one of the unique characteristic of flowering plants. So your stamen is shown here with two lobes of anther attached to the filament. We know that anther is producing pollen grains. To observe the detailed structure of anther, we usually cut anther in transverse section or in horizontal section. On cutting anther in transverse section, we are able to observe the detailed structure of anther. Within the transverse section of anther, we are able to find outermost layer of cells called epidermis. Within epidermis, we find endothecium. Endothecium encloses middle layer. Inside the middle layer, there is layer of cells called tapetum. Tapetum encloses or surrounds sporogenous tissue. So anther being tetrasporangiate, there are 4 sporangia, 1, 2, 3 and 4. 2 in each lobe of anther. So this is one lobe of anther and this is another lobe of anther. These 2 lobes of anther are attached by connective tissue called connective. So all these layers of cells or all these structures which are constituted by different cells can be observed in better view by using microscope, and under microscope we are able to observe different cellular arrangement of these different structures. So this is an enlarged view of a microsporangium as shown here. Over here we are not able to see the cellular arrangement. Now under observing under microscope, we are able to observe the outermost one layer of cells called epidermis, and inside epidermis we have endothecium, which is enclosing middle layers. Within the middle layer we have tapetum, and tapetum is housing the sporogenous tissues. In it we have microspore mother cells, which are also known as pollen mother cells. Microspore mother cells will later form microspores, and the process of formation of microspores from microspore mother cells or pollen mother cells is known as microsporogenesis. I repeat, microsporogenesis is process of formation of microspores inside microsporangium from microspore mother cells or pollen mother cells. Microspores formed will later develop into pollen grains. So technically we can explain microsporogenesis as a process of formation of microspores from a microspore mother cell through process of meiosis. Meiosis is a meiotic division, a reductional cell division, a type of cell division called reductional cell division. So microsporogenesis is process of formation of microspores from microspore mother cells. We have seen that within the tapetum there are cells which are forming sporogenous tissue. Sporogenous tissue is constituted by number of microsporocytes. Microsporocytes are diploid in nature. Diploid here refers to containing entire number of chromosomes of that plant. So microsporocyte is diploid cell, and over here it is shown as gray colored region. And this gray colored region representing microsporocyte is surrounded by a wall. So the wall here is callose wall. So callose wall is surrounding diploid microsporocyte. Once diploid microsporocyte undergoes meiosis, then there is formation of microspore mother cell. So after undergoing first meiosis, there is formation of two microspores from microsporocyte. So after undergoing meiosis 1, there is formation of haploid cells, right? So haploid microspore diet is formed from microsporocyte after undergoing meiosis 1. So these two cell stage can be called as microspore diet. Diet refers to 2. On undergoing meiosis 2, there is formation of 4 cells. So number 4 can be also known as tetrad. So haploid microspore tetrad is formed after undergoing meiosis or completion of meiosis of microsporocyte. So microsporocyte will undergo to form 4 microspores, and these 4 microspores are haploid in nature, and all these cells, 4 microspores, are protected by callose wall. Later there is reorganization of these haploid microspore cells. Along with that, there is also digestion or breaking down of callose wall by callase enzyme. And then there is formation of 4 microspores, and these 4 microspores are microspore mother cells. So microspore mother cell can be also known as pollen mother cells. So pollen mother cell, on undergoing mitosis, then there is formation of 2 cells within the pollen mother cell, and these 2 cell stage can be called as bicellular pollen. So in the bicellular pollen, we have 2 cells. One cell is called as vegetative cell, the other cell is called generative cell. So this is 2 cell stage of pollen. Again, on undergoing mitosis 1 of generative cell, there is formation of tricellular pollen, 3-celled pollen grain. So generative cell will undergo mitosis to form 2 more cells in addition to the vegetative cell. So there is formation of 3-celled cells in the pollen grain. So one cell is vegetative cell, and we have 2 more cells which are formed from generative cell called male gametes or sperm. So all these events are occurring whenever there is sporogenesis occurs inside the flower. So first we have diploid microsporocyte, which is undergoing meiosis, and there is formation of pollen mother cell or microspore mother cell, which later undergo mitosis to form 2-cell stage of pollen or tricellular pollen grain. So let's summarize all these events. First, we have diploid sporogenous tissue, which is constituted by microsporocytes, which will undergo meiotic division and form haploid microspore tetrad, which is shown here. And later there is reorganization and digestion of the callose wall by callase enzyme. Callase enzyme is released by tapetum. So all these are happening inside tapetum. Later there is formation of pollen mother cell or microspore mother cell, which is having potential or ability to develop into pollen grain. So each microspore in the pollen mother cell will be having potential to develop into microspores, which later develop into pollen grains. Development of microspores into pollen grains. How microspores will be developing into pollen grains? For that, anther has to mature. So after the maturation of anther, there is drying up of anther occurs, which can be technically called as dehydration of anther. As the anther dehydrate, there is dissociation or separation of microspores formed within the microsporangium. So within the microsporangium, as the anther dries, there is separation of microspores. After the formation of microspores, usually microspores are associated close to each other. But because of drying up of anther and losing of water, there is separation of microspores inside the microsporangium, which will later develop into thousands of pollen grains. So many thousands of pollen grains are developed within the anther, which will later be released out by break opening of the anther through the longitudinal lines called longitudinal grooves, or which can be also called as line of dehiscence. Dehiscence here refers to break open. So anther break open at longitudinal groove, and there is release of thousands of pollen grains. So this is how pollen grains are released from anther as the anther matures. Pollen grains. Pollen grains are male gametophytes. That is, they are going to produce male gametes. So pollen grains will be producing male gametes. That's why they are considered male gametophyte. So the male reproductive sex organ. Pollen grains occurs in different size. You are shown here with the pictures having pollen grains differing in size. Shapes of the pollen grains are also different as that of the colors of the pollen grain, and the design also are different. So pollen grains occurs in different size, shape, color and design. Pollen grains of hibiscus can be observed with naked eye, and these usually have yellow in color. Whenever we touch the pollen grains of hibiscus or China rose, these are powdery. These are looking similar to the powder. Usually pollen grains are spherical in structure, measuring about 25 to 50 micrometers in diameter. So here majority of the pollen grains shown in this picture are spherically shaped. So spherically shaped pollen grains are having size of about 25 to 50 micrometers in diameter. Pollen grains are having two layers of walls which are protecting the pollen grains. So the outer layer of wall is called as exine, which is usually made up of sporopolinine, a hard material, which is very durable. So sporopolinine is the material which is present within the exine, the outer layer of pollen grain. At some region, sporopolinine is absent. Over here, sporopolinine is absent. Here also shown absence of sporopolinine, and at the regions or at the sites where there is absence of sporopolinine, these are pollen grain used to have pores at the site where there is absence of sporopolinine. And sporopolinine, the exine, the outer covering of the pollen, usually occurs in different design and different pattern. Within the exine, the outer layer of pollen grain, we have intine. Intine is the inner wall of pollen grain, which is present inside the exine, and these are made up of cellulose and pectin. So cellulose and pectin makes up the intine layer of pollen grain. So we have, in total, we have two layers of walls, exine and intine, in pollen grains. Sporopolinine in exine of pollen grains. Sporopolinine of exine of pollen grain is sporopolinine in exine of pollen grains. Sporopolinine is one of the most resistant organic material found as the structural component of outer wall of pollen grain, that is exine. So exine is having one of the most resistant organic material on earth, and because of that, the sporopolinine, there is existence of pollen grains or survival of pollen grains for thousands of years. These are also found as fossils preserved in the earth crust because of the sporopolinine material. So sporopolinine, which is constituting exine of pollen grain, is one of the most resistant organic material which can survive, which can withstand at high temperature, and there is no strong acids or alkaline that can degrade sporopolinine or that can degrade the outer wall of pollen grain that easily. So sporopolinine is difficult to break down, difficult to destroy by strong acids and alkaline. Because of the sporopolinine in the intine of pollen grain, pollen grains are able to withstand at high temperature, and there is no enzyme that can degrade easily, that can destroy easily the sporopolinine of exine. So sporopolinine in exine, the outer wall of pollen grain, is found to be most resistant organic material which can't be easily destroyed by strong acids, strong alkali, and there is no enzyme that can break down sporopolinine. And this is one of the most important questions usually asked in exams. Cells in the pollen grain. We have already seen a stage of pollen grain having 2 cell and 3 cell. So cells in the pollen grains are usually surrounded by cytoplasm and its cell membrane. Now we have bicellular pollen stage, and in the bicellular pollen stage we are able to find a vegetative cell. So vegetative cell is also known as tube cell. Why? Vegetative cell will be forming pollen tube. That's why it is called as tube cell. Vegetative cell is usually bigger in size compared to the generative cell. The nucleus of vegetative cell is irregularly shaped, and vegetative cell have dense cytoplasm. Vegetative cell is also having good food reserve. So abundant food reserve is present within the vegetative cell. Vegetative cell will be nourishing generative cell. So the other cell is generative cell. Generative cell shown here is floating, is present within the cytoplasm of vegetative cell. Generative cell is usually spindle shaped. Two ends are tapered, two ends are thin, and the middle portion of the generative cell is bulged. So it is having spindle shaped structure. generative cell is very small as compared to a vegetative cell. It should be a one eye, not two eye, sorry. So number of cells in pollen grain at the time of release. So whenever pollen grains are released in the nature for pollination, we have different types of pollen grains, and all those pollen grains are not having same number of cells within it. Some pollen grains have, say, like our 60% of the angiosperms usually release pollen grains having two cells. So bicellular pollen grains or two-celled pollen grains are released by our 60% of the flowering plants or angiosperms. 40% of the flowering plants usually release three-celled pollen grain. That is, we have three cells within the pollen grain. Generative cells will divide to form two male gametes. So we have vegetative cell. In it we have already divided male gametes. So two male gametes plus vegetative cell forming three cells in the pollen grain. So we have different types of pollen grains released in the nature for pollination. Our 60% of the pollen grains have two cells, vegetative and generative cell, and in the 40% of the angiosperms, their pollen grains are released at three-celled stage, and three cells are two male gametes plus one vegetative cell. This is just for information. Pollen grains are rich in nutrients. Because of the richness of nutrients in pollen grains has been used as food supplements in the form of pollen tablets or serum. Likewise, pollen grains are also used to increase the performance of athletes, sportsmen, and to increase the speed and performance of race horses. These are benefits, helpfulness of pollen grains. Now, pollen grains can also cause allergy. Different types of allergies are caused by different types of pollen grains. So pollen grains are also allergen. So pollen grains of many different species of plants can cause severe allergies and bronchial afflictions. Bronchial afflictions here refers to disorder, discomfort, disease of respiratory tracts, respiratory tubes. So pollen grains can cause disease related to respiratory system, and that can lead to chronic respiratory diseases. For example, asthma, bronchitis. Bronchitis is inflammation of bronchioles or respiratory tracts. One of the most common example of allergen is Parthenium, which is also known as carrot grass, gajar grass. Parthenium came to India as a contaminant or wheat. Unwanted grasses along with the imported weeds, and later started growing in India, and these are now available almost in all states of India, and these releases harmful pollen grains which will cause asthma like respiratory disorders. So Parthenium is one of the most common example of allergen, especially the pollen grains which are very allergic and can cause allergic disease. So this is harmfulness of pollen grains. Now this is a question: can we store pollen grains like the way we are storing semen of many animals for artificial insemination in future? To answer this question, you need to understand the meaning of artificial insemination. Artificial insemination here refers to transferring of male gametes into the female reproductive tract for reproduction. So, can we store pollen like the way we are storing sperms in the sperm bank? Yes, scientists can store pollen grains in pollen banks. How do scientists store pollen grains for future use? Scientists usually store pollen grains in liquid nitrogen at freezing temperature, say minus 196 degrees Celsius. You're shown here with a fridge opened. As it is opened, there is coming out of vapors. Pollen grains are stored for thousands of years, many years, in liquid nitrogen at freezing temperature of about minus 196 degrees Celsius. So how long do you think the pollen grains can retain its viability? Term viability of pollen grains here refers to existence or survival of pollen grains. So how long pollen grains can survive after its release? There are different types of plants, different types of flowers. So different flowering plants show different duration of viability of pollen grains. Periods of viability of pollen grains are highly variable. Different plants are having different pollen grains, and their survival is different. Say in case of wheat and rice, their pollen grains can survive, can become viable, or these become functional for half an hour or 30 minutes after its release. In case of families of Rosaceae, rose family, Leguminosae, beans, family of beans and peas, and Solanaceae, family of potato, tomato, brinjal, and all. All of these produce different pollen grains, and their pollen grains can survive for months. So viability of pollen grains can differ from few minutes to months. Some even can survive for thousands of years due to presence of Sporopolinine in the exine of pollen grains. So the main factors which are affecting the survival or viability of pollen grains are temperature and humidity. Now with this we have completed the topic microsporogenesis. So you need to answer these questions. First question asks you to describe microsporogenesis. Second, you need to explain the structure of pollen grains with the help of well-labeled diagram. Third, you have to explain how do scientists store pollen grains for future use. Fourth, you need to distinguish between vegetative cell and generative cell of pollen grain. Fifth, you have to give reason why do pollen grains are well preserved as fossils. With this, we have completed basic aspects of microsporogenesis. Thank you. Have a good day.